issue there. Anyway, we can get started. So thank you to everybody who's here in the classroom and lots of people on Zoom today. So I'm going to hand it right over to Dale Laughinghouse, who's going to do the introduction for our speaker today. Thanks for being here, Dale.
Well, thank you so much. I'm also getting emails about from the people wanting to attend, and they are getting asked for a passcode. So is there a way? Is it just that you have to log in as an outside user, right? Is that what the people that are not UF?
Oh, is it saying like sign into the host kind of thing?
Correct.
Yeah, yeah. There should be a little link underneath that, just saying join as an attendee, and then you won't be prompted for a passcode or anything like that.
Okay. So I'll do that, yeah, in a second. But I'm not going to try to multitask and present and introduce Forrest and try to email at the same time, since I might forget something, get messed up here. But anyways, so thank you everyone for coming to this seminar. It's my pleasure to introduce Dr. Forrest Lefler. So Forrest started in my lab as an intern. What is it, Forrest, how many years ago? 6 years ago? Yes, 6 years ago.
Yeah.
He had started, he had worked initially with Dr. David Berthold in Dr. Gantar's lab at FIU. And from there, he came over to start as an intern. He really didn't want to do, I asked him to do a grad school, if you want to go to grad school. He's like, no, I don't really want to go to grad school. I don't want to. So I asked him again. And then later on, he decided to go to grad school. He decided to start to do a master's degree. And 1 year along in his master's degree, he said, he goes, I want to do a PhD. And I go, why? He goes, there's so much to do, but so little time. And I said, well, that's not good enough. So anyways, so he went back, he gave me a better answer. And so, he finished his PhD. If I remember, he published 15 papers during his PhD, which was wonderful, very, very prolific writer, both author and co-author and an author. And now he is a postdoc in the lab as well. It's kind of hard to get rid of him, but he's a pleasure to have around and does great work. So today he'll be talking about the diversity of cyanobacteria and in tropical and subtropical freshwaters. So our blue-green friends, like Nicole Petrasius likes to say, in both Belize and in Florida. Part of this was his PhD, and part of it is ongoing research he does in the lab now. So with that, Forrest, I'll give you the floor, and I will write a couple people back that are having difficulty getting in.
All right, thank you. It's my favorite story. So I'm Forrest, and here today to talk about cyanobacteria. Let's get right into it, because I have more slides than I have time probably. So I'm just going to give an outline, general introduction on what cyanobacteria are, how we can assess their diversity. I'm going to talk about CyanoSeq, which is a database of cyanobacterial sequences that I created during my dissertation, and also spatial diversity along the New River in Belize, and spatial temporal diversity within the Kissimmee chain of lakes and Lake Okeechobee as two separate projects, and then just a general conclusion. So let's get right into it. So what are cyanobacteria? Cyanobacteria are oxygenic photosynthetic bacteria. These evolved around 3.8 billion years ago, and we know that from stromatolites, which are fossilized microbial mats. And just to give context, eukaryotes evolved around 2 billion years ago. And cyanobacteria are really interesting because these were absorbed by eukaryotes via symbiotic events, and they became the chloroplasts in those in plants and other algae. And so you can see here's a phylogenomic tree of all of the other cyanobacteria, and you can see here these plastids are nested right in there in this group. They're primary producers. They can fix atmospheric carbon and nitrogen, and they can also form blooms, which can produce toxins and taste and odor compounds. And these are going to be more of the focus of what we're talking about, what I'm talking about today. Let's see. Oh, Zoom doesn't like me today. Cool. All right. So here's just a general overview of what some cyanobacteria look like. I cannot possibly show all of them, but these are some common bloom formers over here on the right. This is Microcystis, a few different morphospecies, morphotypes, whichever term you prefer. There's also Sphaerostromopsis, another type of Microcystis here. These are 3 different morphotypes of Dolichospermum. This is a Microcystis bloom that was out in Lake Okeechobee. This is Raphidiopsis. I'm going to be talking about these today. And these are just some picocyanobacteria. As you can see, they're very small. They're about 2 microns wide. Here's more picocyanobacteria and just some other non-bloom formers that are also really beautiful. So what are harmful algal blooms? Harmful algal blooms are a dense growth of algae. This can be cyanobacteria, which is of course what I'm talking about today, but this can also be diatoms and dinoflagellates. And they can grow in various areas in the water column. You can have them near shore, you can have them a little further out, you can have them collecting at the surface, dispersed through the water column, maybe at photic depth, maybe well dispersed. So they can form all throughout. They can also form on benthic surfaces, which I won't be talking about much today, but they are there. In the case of cyanobacteria, they're capable of producing several different types of compounds. It really just depends on who the main players are. And they can also contain taste and odor compounds, which smell pretty terrible. And so some cyanobacteria, in the case of, let's say, Microcystis or Deligospermum, have a competitive advantage because they can form gas vesicles within their cells, which you can see here. And this allows them to float up to the surface, which gives them more access to sunlight. This surface area or this little region on the water column is probably warmer too. So they're able to vertically migrate up. And that's when you really get these dense blooms. This is a Microcystis bloom in Lake Okeechobee. And cyanohabs are typically thought of as a freshwater phenomenon, but recently we've been seeing benthic cyanobacterial habs on the Gulf Coast of Florida, and you can see this is a lot of dislodged mat material that's floating out in the ocean. I don't know why my arrow key isn't working today, so that's fun. There are a lot of cyanotoxins. I'm only going to talk about 4 today, but there are a lot more than that. The most common one that if you've heard of any, you've probably heard of microcystin and probably heard of microcystin LR. This is going to be over here in the top right. There's about 256 different congeners of microcystin LR. This is a hepatotoxin, which has been linked to human death in Brazil and maybe more. And this was characterized from microcystis, so microcystin, microcystis. You're going to see a pattern here. There's also nodularins, which is going to be this one here, pretty similar structure to microcystin. It's also a hepatotoxin, and nodularins were characterized from nodularia. Cylindrospermopsins look a little different than those two. This is also a hepatotoxin, which has been linked to human death in Australia. And this was characterized from Cylindrospermopsis, which is now Rhophidiopsis. And then there's also anatoxins, which are a neurotoxin. This is a very different, much smaller. And these were characterized from Anabina, which is now Deligospermum. So drivers of cyanohabs, there's abiotic drivers, and we'll talk a little about the biotic drivers here in a second. But I just want to focus, there's 2 main ones. There's eutrophication, which is going to be an increase in nitrogen, phosphorus, and more, but we're just going to focus on nitrogen and phosphorus today. And this can be from various sources. This can be fertilizer as agricultural urban fertilizer, sewage, septic tanks, industrial effluents, livestock, municipal wastewater, and probably more. And you can see here, this is a limno curtain that was put in a lake. This is a study by Schindler in 1974. And they, on one side, they dumped in a bunch of nitrogen. I'm not sure exactly how that happened, but they dumped in a bunch of nitrogen over here. And on this side, they dumped in a bunch of nitrogen and phosphorus. And as you can see here, this is a lot greener on this side when you have both of them together. And that's because cyanobacteria really like the combination of nitrogen and phosphorus. It's not necessarily just one or the other. But they also like increased temperatures. Blooms like it hot, and this gives them a competitive advantage over other phytoplankton, eukaryotic phytoplankton. And the warmer it gets, cyanobacteria, such as like microcystis, get really, really happy with that increase in temperature. They also have really complex relationships with bacteria and maybe more, but bacteria are probably the best understood in this area. And this is like kind of the concept of the interactome, which is where several organisms work together to help each other fill in genomic gaps, such as biosynthesis of vitamins, assisting in nutrient cycling. And so we can see here, this is a Deligospermum colony here, and you can see it has a mucilage. It's not picked up very well by this picture, but you can see it's absolutely covered in bacteria, which are going to be these smaller ones here. And so these can help each other where Deligospermum is producing carbon and oxygen for these bacteria. And in turn, they're probably producing several other things, maybe helping in phosphorus cycling, urea breakdown, as well as vitamin synthesis. And this is pretty well described in the case of microcystis, where there have been metagenomic studies done looking at microcystis and also looking at the associated microbiome and the functions, the genomic functions that these have. And as you can see here, there are a lot more genomic functions contained within the microbiome than just within microcystis. So they form very intricate relationships that are very important. But cyanobacteria don't just form blooms. I think they are getting a bad rap lately due to all of the blooms that are happening. But they're very important and they're all over the place. They're out in open oceans. They're in hypersaline pools, polar areas, desert soil crust. If there is water and if there is light, cyanobacteria are going to be there. And the classification of cyanobacteria, getting into more of the diversity aspect, dates back to Linnaeus, who described Tremella nostoc, which is now Nostoc commune. And there have been several revisions since then. Probably most importantly is in 1970s is when they were really confirmed to be bacteria. And they're a monophyletic class of bacteria that's going to be the Cyanophyceaceae. And there have been two recent revisions in the last decade or so, and that's going to be 2014, where the focus really began on the genetic relationships using the 16S rRNA to understand who is related to whom. And there was just a recent revision from Strunetsky that came out last year, or late last year, that focuses on the phylogenomic relationships between these taxa. So how can we assess the microbial diversity of these? Probably the most common way, and dates back to microscopes. The invention of microscopes is microscopy. But this can be really difficult to identify and requires a lot of taxonomic expertise because a lot of cyanobacteria look alike but are evolutionarily distinct from each other. So for example, right here, these two things are completely different genera. This is the Legospermum up here, probably Circinalis, and this is Sphaerostromopsis torquus reginae right here. These are two different genera of cyanobacteria, and if you didn't know exactly what to look for, which is going to be this little cell right here, or if it didn't have that cell, it would be really difficult to distinguish these two. And we also see here, these are three different genera of cyanobacteria that don't look too terribly different. And so that makes it difficult to characterize these via microscopy. There's also picocyanobacteria, which again are like 2 microns large, and so they're very hard to delineate those taxa and also enumerate. And if anybody has ever done phytoplankton counts, I can guarantee it's not their favorite thing to do. It's very time intensive, very laborious, and it's just overall not fun. There's also clone libraries in DGGE, and these are really good methods, but they can't capture the full diversity of whatever system you're studying. And then there's metabarcoding, which is a more new method, which has come with high throughput sequencing. And this is really ideal for diversity studies, but there are also several caveats to this. It's not perfect. And metabarcoding relies, again, on next generation or high throughput sequencing, sequencing these samples and getting sequences. In the case of cyanobacteria, it's typically the 16S. In the case of bacteria and archaea, it's also the 16S, but there are other markers that you can use. But again, this isn't perfect. This relies on PCR, and so you can have primer bias, which can affect your results. But it at least gives us a 16S fragment that we can place on something to give us information on who is there. And this is really important because this really relies on a taxonomic database to assign your reads. Because if you have a sequence and you don't know who it belongs to, that sequence isn't very useful, right? Which brings me into the first chapter of my dissertation, what I'm going to be talking about today, which is Cyanoseq, which is a reference database of cyanobacterial 16S rRNA sequences with a curated taxonomy, and that is a long title. This is in press in the Journal of Ecology, and this is a QR code to the database, but I'll have it again at the end. If you're curious, you can also email me if you are curious as well. We created this in the lab because commonly used databases for taxonomic assignment of environmental reads, such as Silva or Green Genes, lack much of the work for cyanobacterial taxonomic works. There are cyanobacterial genera being described frequently, and they're not making their way into these databases, which then causes a problem because you don't have a good taxonomic database. And this leads to a problem because microbial ecologists who are conducting their diversity analysis, such as us, are undercharacterizing the cyanobacterial diversity. And so this is an example of the problem. This is really where where Cyanoseq was born out of some data that we got. And so, like I mentioned earlier, on the Gulf Coast of Florida, they are experiencing these benthic cyanobacterial mats. And we have people in the lab who have been working on these since 2016, isolating, characterizing, describing, and even publishing new genera from these mats. So we had a pretty good idea of who should be in these mats. And when we conducted our metabarcoding studies and classified with Silva, because Silva is the most commonly used, we did not find them. Okay, we were expecting to find Dapis, Okeania, and Serena capillaria because, again, we have isolated, characterized, and published on isolates from this. But we found Trichodesmium and Planktothrix and Formidium in these samples. And this is a problem because Trichodesmium, it doesn't really belong here. We know where Trichodesmium occurs, and that's going to be out in the open ocean forming these large blooms. This is a satellite image of, I think, the Indian Ocean, but one of the oceans, one of the warmer oceans. And so it didn't really make sense to find them in here. And also Planktothrix is going to form blooms in fresh waters, not in the Gulf Coast of Florida as a benthic mat. But however, when we use cyanoseq to characterize these, we do get Okeania, Adapta, Serena, Capillaria, and a few other ones which have been described in marine benthic mats and marine systems throughout the world, whereas opposed to getting some other taxa that aren't necessarily informative. So that's just an example. But why should you care about taxonomic assignment? Because it can help us understand the ecology and the diversity of the systems that you're studying. It can help you, one example is to identify potentially toxigenic genera. Going back again, if we would find that we have Planktothrix in here, which is potentially producing microcystins or saxitoxins or who knows what else. But that's not the case. We don't have Planktothrix, it's not there. And the ecology and habitat of many of these taxa are well described. People have been doing cyanobacterial taxonomy since Linnaeus, so as old as taxonomy itself. And then also just finding Planktothrix and Trichodesmium in that case together in a marine benthic mat just doesn't make sense. But if you didn't know that, you wouldn't know what else to base it off of. And this can help for more accurate diversity assessments because eDNA is being used for management. This is becoming increasingly common, and I know the Cyanobacteria, or the California Cyano Working Group has started employing these in some of their water bodies across the state. And it's important to understand who is there, and because that can help you again understand which toxic genera are potentially there. And also, as a taxonomist, I have to stress this: these names are important, right? And there are a lot of other databases that exist. Silva is one I mentioned, but there are several more, and these all have a lot of problems in regards to the cyanobacteria. But when it comes to things like Silva and green genes, I can't speak to their bacterial side because I am not a bacterial taxonomist. So just quick conclusion, it can be used to better characterize cyanobacterial communities. This is the foundation for the taxonomic assignment throughout the rest of what I'm going to be talking about today. It can be used with common pipelines such as QIIME2, Data2, ID taxa, and it's available here. And this will be at the end of the presentation as well. So cool. That's enough about taxonomy. Let's get more into diversity. So this first topic I'm going to be talking about is in the New River. And this was done in 2019. And so just background on the New River, it's the longest river confined entirely to Belize. It begins in the south in the New River Lagoon, and it flows north and empties into Corozal Bay, which is going to be the Caribbean Sea. It's a very slow moving river, and it has several uses. People live directly on this river, as you can see, and it's a very vital water source to the community. There is a lot of land usage along the river. There's agricultural usage, industry, urban, and aquaculture. So you can see here, here's a shrimp farm right on the river. This is a sugarcane factory, which processes sugarcane and turns it into sugar. And you can see where the effluent from this comes and drains directly in the river. This comes from an urban area. I'll give you a guess what that is, but it stinks. And also this is just other effluent. So there's a lot of stuff happening on this river. And in 2019, when the study occurred, Belize has a warm tropical system, somewhat similar to Florida in the fact that they have wet and dry seasons, right? And in 2019, they had a prolonged drought that lasted well into their rainy season. So they depend on the rainy season to come and flush this river. But it didn't come, these rains never came, and this already slow moving river started moving even slower. There was a concentration of nutrients and pollutants in the river. And there were reports of algal blooms as well as associated fish kills and the stench that comes with both of those things. And so we've been down there, and our objectives were to characterize the cyanobacterial diversity, and because there were reports of blooms, determine which, if any, bloom forming taxa were present. And also to understand the changes in community structure due to this various anthropogenic influences that occur on the river. And so to do this, we conducted sampling at 29 sites along the river, starting in the south in the lagoon and working our way up north out into the bay. There's a high concentration of urban area here. There's a lot of agricultural happening here, agricultural activity here. And then they're down here more towards the mouth of the river is where the aquaculture is. We collected samples from microelements, chlorophyll, as well as eDNA for barcoding. We used two different primers here. Today I'll be talking about the cyanobacterial specific primers. And our metabarcoding was done on Illumina MySeq, and we processed our reads with Data2 and assigned taxonomy using Cyanoseq, and we did a lot of other bioinformatic magic. Okay, I'm not going to bore everyone with that, but if you're curious, please ask me. And what we found, even in areas with higher chlorophyll, is that they were dominated by the prochlorocaceae, which we really did not expect. When the prochlorocaceae are small, Cyanobacteria, those are going to be those less than 2 microns. So this is really not what we were anticipating. We were anticipating maybe some Microcystis and maybe some Dolichospermum Rhophidiopsis, common bloom formers, especially in warmer climates. Now, we did see a lot of Microcystis throughout the river, but it never dominated except for maybe like 2 samples. It was really these Cyanobacteria that dominated, but they were also very diverse throughout the river as well. This wasn't a single Cyanobacterium that dominated. There were several. There were dozens that dominate or were found throughout the river. And so you can see starting down here, which is going to be here in the lagoon, you have a dominance of this ASV, which is going to be Amplicon sequence, this type of sequence. And we can see it's pretty diverse in the urban and agricultural areas. And we see the diversity shifts as we get more towards the mouth of the river. And now just backtracking a little bit on our Cyanobacteria, these are found everywhere. It's not shocking that we found them. It's more shocking that we found them to be so dominant. These are tiny, often unicellular. This is what you're going to see under a microscope, and it's really hard to tell the differences between them. This is not what you're going to see, despite how cool this looks. That's not the reality, but it looks really cool. This is going to be, I think, a scanning electron microscope. And these are mainly within the Cynococales and the family Prochlorocaceae, although there are some Cyanobacteria that exist outside of that. And these are really difficult to identify. There are a handful of different Cyanobacterial genera in these photos, but you can't really tell. Also, there's not a lot of defining morphological features. And even then, using metabarcoding has its limitations for these because the 16S RNA is highly conserved within this family. So it makes it quite difficult. And oftentimes you have to rely on phylogenomics to delineate these taxa. However, we did find quite the diversity of these. And we did find that if the anthropogenic influences did lead to an effect on the Cyanobacterial community, it did lead to an effect on the overall cyanobacterial community, but I'll only be talking about the Cyanobacteria today. And we see here in areas of agricultural and urban influences, you see that they're clustered pretty close together. They were not very different from one another. But once you got more towards the mouth of the river, where there was aquaculture and also some saline salinity increases, we did see that the Cyanobacterial community shifted then to a dominance of this, which ended up being an interesting species of Volcanococcus, if you're curious. And then out in the more out into the bay, we saw a dominance of this, which was Parasinicococcus, which makes sense. This is known to be in more warm coastal environments, right? And we did see that there were differences in the alpha diversity between these some of these communities, where we saw that the diversity was highest in agricultural and urban industrial areas, and lower in aquaculture, coastal impacted, and unimpacted areas. And I should have mentioned earlier, unimpacted areas are going to be our lagoon. There's not a whole lot happening up there. It is quite beautiful. But one thing that's was a little shocking to us is this dominance of Cyanobacteria is really interesting because they're known to form blooms in warm freshwaters. And again, we're in Belize, we're in the tropics. These are warm freshwaters, and they're known to form blooms in Africa and their Great Lakes, southern Spain, Mumbai, Southeast Asia, particularly Singapore, where recently there was a Cyanobacterial bloom, which produced 24.6 ppb of Cylindrospermopsins, which is this toxin right here. This is a hepatotoxin. So that's really interesting. We went through, and I know this is a big phylogenetic tree, but let's just look at the pretty stars. This is really interesting because a lot of the ASVs that we collected from Belize did cluster with some known Cylindrospermopsin producing taxa. However, we did not find Cylindrospermopsin in our samples, so it may be below detection limit. We can't rule out that they're there, but we can say there are likely some potential Cylindrospermopsin producing taxa in the New River. So just to wrap this up, there was some Microcystis, not a lot of Nausicales, which we're going to talk about here in a second in these coming topics, which was surprising to us because Nausicales and Microcystis, these are known bloom formers. These are really what we anticipated, but they really weren't there. These Cyanobacteria may be able to form blooms. We saw, these high chlorophyll areas dominated by Cyanobacteria, and they may be producing toxins. And this can affect, again, these people live right on this, right on the water. This is a source of drinking water, and this can impact their drinking water. And it could potentially be a problem because the effects of chronic exposures of cyanotoxins are really unknown. But most importantly, to me, I guess it's important, right, is the anthropogenic influence has really affected the cyanobacterial community, both the total cyanobacteria and the Cyanobacteria, which is pretty cool. So coming closer to home, let's talk about the regular, I guess, cyanobacterial community throughout the Kissimmee chain of lakes. And this is a 3-year-long project. We're currently in year 2, and I'm only showing the data from year 1. So there is not a lot of data here, but there's a lot more work to be done, and there's some interesting data. And if you're not familiar, the Kissimmee chain of lakes is up here in the Orlando-Kissimmee area, which works its way down and flows through the Kissimmee River and drains into Lake Okeechobee. These are really the headwaters of Lake Okeechobee and the overall Everglades. And the point of this project was to assess the phytoplankton community near hydrilla beds to search for Autochthonous hydrolicola. And Autochthonous hydrolicola is a recently described cyanobacterium that produces a toxin, autochthonatoxin rather, which means eagle killer, or toxin which kills eagles, because this is known to bioaccumulate up the food chain and lead to deaths in bald eagles. So not very good. So we didn't find it, which is kind of bad for the project because we were looking for it, but also good because it leads to deaths in eagles. So it's kind of a bad thing. But we did find other bloom-forming cyanobacteria, which is really cool to me because they're a little different than what's in Lake Okeechobee, and we'll talk about that later. But that's also kind of bad because they're bloom-forming cyanobacteria, right? We wanted to look at the differences in the cyanobacterial communities between these lakes. These are all interconnected. Maybe not connected with each other, but they're at least connected in some way. And we wanted to look at drivers of community structure throughout these lakes. And to do this, we sampled in 7 lakes. We took 3 samples per lake to try to get a representative sample. And this was done 3 times during this first year. And that's all those are the data I have today. We collected a lot of physiochemical parameters, as well as eDNA for metabarcoding of bacteria, including cyanobacteria. We also collected for protists, but I won't be talking about that today because I don't have time. And what we saw was that it didn't matter which lake that we were looking at. Here's each of the 7 lakes. These communities were dominated by diazotrophic cyanobacteria. And these lakes were often P-limited, at least based off the current data that we have during the times of our sampling. And we saw many bloom-forming cyanobacteria, such as Planktothrix, Deligospermum, which used to be Anabina, Microcystis, Aphanizomenon, Raphidiopsis, which used to be Cylindrospermopsis, but also Cuspidothrix and Chrysosperm. So lots of bloom-forming cyanobacteria, lots of different bloom-forming cyanobacteria were found throughout these lakes. And in comparison to Belize, there was a lot lower relative abundance of Cyanobacteria. And the Cyanobacteria are going to be here in this darker blue. You can see there's overall not a lot. Sometimes there is a lot though, right? And we saw that there were differences between the bloom formers in each lake, where we can see here in Lake Tohopekaliga, there seems to be a lot of Sclerospomopsis, which is found throughout the year. But in the Cypress, in our very first sampling period, there was a lot of Aphanizomenon, whereas East Lake Tohopekaliga had a lot of Planktothrix. So really interesting. Also really interesting in Lake Istapoga, there seemed to be a lot of Wolea, which isn't really a bloom-forming cyanobacterium, but closely related. And we conducted an NMDS and looked at Permanova and a bunch of magical multivariate stats. And we saw that there were differences between these cyanobacterial communities as a whole. Now, not every lake was different than every other lake, but for example, the communities that we sampled in the northern region of Lake Okeechobee were different than Lake Cypress and East Lake Tohopekaliga. And we can see that makes sense. Because the colors, we're just really looking at colors here, right? Because that's the relative abundance of each of these taxa. And so you can see here, Lake Istapoga right here has a lot of Wolea, and that's very different than, say, Lake Okeechobee, which is right next to it, which has a lot less Wolea, a lot less Aphanizomenon. So the communities are distinct from one another. But again, it's just certain lakes that are different from one another, and some lakes are a little bit more similar than others. But again, our focus was to sample near hydrilla beds to see if we found Autochthonous hydrolicola. But interestingly, looking at the communities between the samples that were collected on top of hydrilla beds or next to hydrilla beds, as opposed to away from hydrilla beds, we found that near these hydrilla beds, there was a higher abundance of bloom-forming genera, such as Cuspidothrix, which is one of the smaller ones here. Don't try to find it. But also Planktothrix was a lot higher. So you can see near hydrilla beds, this darker green bar represents Planktothrix, and there's a lot more here than there are when hydrilla is not present. Also Raphidiopsis, you're going to see more of, which is going to be this light yellow bar. There's more Raphidiopsis in the presence of hydrilla. So hydrilla seems to be having an effect on the cyanobacterial community structure. It also has an overall effect on the bacterial community structure as well, which we found really interesting because it seems like hydrilla has an overall effect on the bacterial plankton, including cyanobacterial community structure. And again, this is just year one, so I don't have a long-term spatial temporal data set to really work with here. But we do see that there seem to be different water quality parameters driving some of these bloom-forming cyanobacteria. And some of these bloom-forming cyanobacteria don't really like each other, where we see here like Sphaerostromopsis and Aphanezomenon don't really like to coexist together. And Aphanezomenon seems to like orthophosphate and nitrites. Nitrogen is hard. But we need more data to really evaluate the drivers of these various bloom-forming cyanobacteria and determine why certain lakes have Planktothrix, why certain lakes have Aphanezomenon, et cetera. So we're hoping we're able to answer that at the end of this project. Which I kind of gave away some of my conclusion slides here. Lots of bloom-forming cyanobacteria present in these lakes, and they seem to dominate the cyanobacterial community. Now, I do want to stress this is just the cyanobacterial community. This is not the phytoplankton community. I am by no means saying that bloom-forming cyanobacteria dominate the phytoplankton community of Lake Kissimmee, but they do seem to be dominating the cyanobacterial community. The species richness increased as we moved down. It was lowest in the northern parts and increased as it worked its way down into the Lake Kissimmee. And it was actually highest in Lake Okeechobee, which is interesting because it flows down. And the planktonic community seemed to be influenced by hydrilla, both the bacterial and cyanobacterial community. And one thing we're looking at is how other aquatic plants seem to influence these communities as well. Is it just hydrilla that leads to an increase in some of these bloom formers, or maybe I'm drawing a blank on some of those other SAVs, but maybe Naiad has an effect on cyanobacterial communities. And again, this is just the results from the first year from a multi-year project. But getting into Lake Okeechobee, looking at the spatial temporal diversity of Lake Okeechobee. Lake Okeechobee is at the end of the Kissimmee chain of lakes here. It's a pretty large lake. It's a large eutrophic, shallow subtropical lake. It's a mouthful. And it's pretty shallow. Its average depth is only about 3 meters, but it depends on how much water is pumped in or out of the system. And it has garnered attention in the last several years because there were microcystis blooms in 2016 and 2018, and the one in 2018 covered about 90% of the lake surface. And both of these were producing microcystins at various concentrations. So it's been a hot topic. And so blooms aren't a new phenomenon in Lake Okeechobee. They've been recorded since at least the 1970s, as far as I can find. But they do seem to presently be dominated by Microcystis and Endoligospermum. There are also older reports of Aphanezomenon blooms and Raphidiopsis blooms in the system as well. And we do see Raphidiopsis blooms occurring now. I've sampled those before, but they seem to be a lot less intense and a lot less frequent than those other two. But the objectives for this study was to assess the spatial temporal cyanobacterial diversity within the lake, identify potential drivers of the cyanobacterial diversity. Today, I'm going to focus on Endoligospermum, Microcystis, and Raphidiopsis, and also assess the spatial temporal diversity of cyanotoxins as well. And so to do this, we sampled at 6 sites across the lake at areas of outflows, inflows, and also on the southern part of the lake. And there were 10 sampling outings that occurred over the course of a year. We collected a lot of physiochemical parameters, including cyanotoxins, and again, eDNA for barcoding, metabarcoding. And for this, I'll be talking about the general bacterial primers. Also, additional data was collected from the South Florida Water Management District's DB Hydro database. Let me let this guy in. So there are a lot of results here. We have a lot of data, and I hope that I can finish in time. But the lake was frequently nitrogen or co-nutrient limited, with P limitation occurring only one time. There were several bloom-forming genera, and there's some potentially novel diversity within the lake, which is really interesting, especially to me, the taxonomist. And communities at the Kissimmee mouth, and so that's where the majority of the inflow is. I think it's something like 70%, but please don't quote me on that number, of the inflow into Lake Okeechobee comes into this area. And the cyanobacterial communities in this northern region of the lake were different than the rest of the communities. And that's probably because this is a source of nutrients and it was associated with an increase in microcystis relative abundance. We found several toxins throughout the lake. We found 2 different congeners of microcystins. We also found nodularins and anatoxins, but these weren't bloom samples. These were just regular surface grabs. And the concentrations were still fairly low. They were about 1 ppb. The highest was nodularin at like 1.2 ppb, so still fairly low. And to our knowledge, at least we haven't been able to find anything. Nodularins haven't been recorded in Lake Okeechobee, but if you have information on that, please reach out to us because it's really interesting to me because we're not entirely sure who the producer is. Please work. There we go. All right, so again, lots of pretty colors, lots of different cyanobacteria found throughout the lake. These are going to be our different sites, our 6 different sites. But what's important is that this system, a little bit more similar to the New River in Belize, is that it was dominated by picocyanobacteria. And this is going to be these light blue bars, light green bars, and pink bars. But there was also high abundance of Deligospermum, which are going to be these dark blue bars you can see here in the southern part of the lake. On our last sampling outing, Deligospermum dominated the cyanobacterial community. And this it was found throughout the lake. But microcystis was also frequently found. That's going to be these dark green bars here. And you can see here, this is at the mouth of the Kissimmee River. You can see it was always there at different relative abundances. And it seemed to be increasing as we ended our study. But also found down in Clewiston where it's, I would say it's dominant within the cyanobacterial community. We did also find Raphidiopsis, which is going to be these orange bars here, but not at a high abundance like we found microcystis or Deligospermum. There was also some Cuspidothrix and such, lots of diversity within Lake Okeechobee, but primarily picocyanobacteria. So just touching on some of the data, again, I don't have time to cover everything, but we have wet and dry season here in Florida. Luckily for all of us, or for some of us, it doesn't snow here, which is great. But we do see that there is a difference between the communities in the wet season, and those communities seem to be more dominated by Deligospermum, which you can see here in the light green, whereas the dry season seems to be more have a higher relative abundance of Raphidiopsis. And these communities do differ from one another based off of beta diversity. But when it comes to alpha diversity, they're not significantly different from one another. So it seems like the communities change, but there doesn't seem to be less or more of cyanobacterial abundance. So let's get into drivers. Okay, blooms like it hot, right? Microcystis seems to like it the hottest. Okay, so this is going to be temperature down here, and you can see increasing temperature. It seems to be cut off a little bit. I need to work on that. I always do something like that, but around like 30, 31 degrees Celsius, we see an increase in microcystis relative abundance. And these stars mean that it's significant based off of the generalized additive model. Whereas we get more warm temperatures in Raphidiopsis, we see a decrease in Raphidiopsis relative abundance. Whereas Deligospermum doesn't really seem to care too much about the temperature, which is really interesting. But the sun also produces light. Light goes into the water column, and Deligospermum and microcystis both like a very high photic depth. So we can see here, as there was increasing photic depth, there was an increase in microcystis and Deligospermum relative abundance, which was interesting because Raphidiopsis had the opposite effect, whereas there was an increase in photic depth, there was a decrease in Raphidiopsis relative abundance. And so that was pretty interesting. Another interesting thing was this is microcystis in the dry season, which is our cooler season, our less hot, still hot. And there seems to be a peak in the microcystis relative abundance in the dry season at about this is like 23 Celsius. So so pretty interesting. There's a bimodal response on microcystis with the temperature. And then also nitrogen, phosphorus are known abiotic drivers of these taxa. And so looking at them independently, we see here that total reactive phosphorus has a negative correlation with Raphidiopsis and microcystis, but these were non-significant. Whereas Deligospermum really likes inorganic forms of phosphorus. It had a linear, positive, significant, all of the fun words response to phosphorus. Whereas nitrogen, it had a negative but non-significant response to dissolved inorganic nitrogen, which makes sense because the Legospermum is a diazotrophic genus. It can fix its own atmospheric nitrogen. It doesn't really need exogenous like nitrates, nitrites, or ammonium. Microcystis didn't really seem to care about nitrogen, which is I found interesting. But what I found interesting was that Raphidiopsis, another diazotrophic genus, had a negative correlation to dissolved inorganic nitrogen. So as this one increased, as dissolved inorganic nitrogen increased, Raphidiopsis relative abundance also seemed to significantly decrease. So that's interesting. But also looking at the nitrogen and the phosphorus ratio, which is something that seems to be driving different bloom forming taxa and driving them differently. We do see that Microcystis likes a high N to P ratio, and this is going to be dissolved inorganic nitrogen to dissolved inorganic phosphorus. We can see here it was significant and positive with Microcystis, but also significant and positive with Raphidiopsis, which was kind of shocking because again, this is diazotrope. And it was negatively correlated to increases in dissolved inorganic nitrogen. Whereas the Legospermum was negatively correlated to increases in the NP ratio, which is interesting because it seems to be, the NP ratio seems to be affecting these two different diazotrophic taxa in different ways. Okay, but since Microcystis and Raphidiopsis both seem to like this increase in N to P ratio, I was curious as to why we really see Microcystis blooms seem to be more frequent and more intense, whereas Raphidiopsis blooms are a lot less frequent and a lot less intense, although they still do occur. And one major difference between the two is going to be photic depth, which we talked about, again, increase in photic depth, decrease in Raphidiopsis, whereas increase in photic depth, increase in Microcystis. But also the overall lake level seems to be having some effect on Raphidiopsis, where this is significant, but I forgot to put my star, where there is an increasing lake depth, there seems to be a decreasing relative abundance of Raphidiopsis, whereas Microcystis is pretty ambivalent to lake depth. So this is interesting because it seems like these two, despite having the same response to these NP ratios, have different niches within the lake, whereas Raphidiopsis prefers more shallow and turbid conditions, whereas Lake Okeechobee doesn't whereas Microcystis doesn't have a preference to lake depth in Lake Okeechobee, but prefers increased photic depth. And I had mentioned these core bacteria that occur with these. And what we saw was that the Legospermum had some uniquely correlated bacteria to it that were distinct from the bacteria that were correlated to Microcystis, which you can see here. I probably should have highlighted these. But they had distinct phycospheric or associated bacteria. And also the Legospermum was correlated with this, which seems to be Neocylindrospermum. And Microcystis had a lot of other cyanobacteria that it seemed to be correlated to, and also the ratio of nutrients as well. Raphidiopsis also had some interesting relationships to bacteria and cyanobacteria, and actually surprisingly shared a lot of those correlations with Microcystis, but not with the Legospermum. But it is a giant mess. It's a very uninformative figure. So I guess I just have to say, trust me, and you'll see it in the paper whenever that gets out. So to wrap this up, there was a high relative abundance of picocyanobacteria in Lake Okeechobee. Legosperma, Microcystis, and Raphidiopsis had distinct drivers within Lake Okeechobee, whereas Microcystis really likes it hot, likes a lot of light, likes a high NP ratio, whereas the Legospermum doesn't really seem to care about the temperature. It just likes a lot of phosphorus and a lot of light. And Raphidiopsis is a lot different. It likes it cooler, more shallow, more turbid with a higher NP ratio. And this is just anecdotal, but I tend to see more Raphidiopsis in the dry season. We did see smaller blooms that occur during the dry season. And that also makes sense because it likes these cooler temperatures as well in the dry season. Again, is more associated with cooler temperatures. There are distinct associated bacteria with each of these bloom-forming cyanobacteria. And nodularins, again, recorded in Lake Okeechobee to our knowledge. This is the first time, and we're still trying to pinpoint who the nodularin producer is within Lake Okeechobee. There were also microcystins. I feel like I skipped over this earlier. There were also microcystins found, but that was pretty clearly correlated to Microcystis. And so overall takeaways, I'm sure I'm getting close to the end of time here, is that CyanoSeq can help properly characterize cyanobacterial diversity. Cyanobacteria appear to be highly abundant in subtropical, tropical waters, particularly in Lake Okeechobee and the New River, but not in the Kissimmee chain of lakes. And we're still trying to deduce why. We're hoping this a longer, more temporal data set from the Kissimmee chain of lakes can give us better insights into this. There are several potential toxin producing taxa of Cyanobacteria found within the New River, and so there may be cylindrospermopsins being produced by them, but also within the lab, we've isolated one of these Cyanobacteria, and we have confirmed that it's producing cylindrospermopsins, but at a very, very low concentration. But this is a really understudied area and really requires a lot more attention to understand really what these mean, right? And if they're abundant and why they're producing toxins. And these Cyanobacteria appear to be more diverse in Belize compared to Lake Okeechobee, but more work and data are needed to really assess this, because again, this data set from the New River was just one sampling that occurred on the river, not a temporal data set. But going back to the bloom formers, there seems to be distinct drivers of these bloom forming taxa in Lake Okeechobee. Again, photo depth, NP ratio, phosphorus all seem to be affecting these somewhat similar but somewhat different. Whereas Lake Okeechobee has a higher relative abundance of Deligosperma, Microcystis, and Raphidiopsis. The Kissimmee chain of lakes is a lot more diverse in the sense of its bloom formers. It has Aphanizomenon, Deligospermum, Planktothrix, Ferrospermopsis. I'm missing a few more here, but there's a lot. And future work is really going to be focused on identifying the physiochemical drivers of these genera in these lakes, because we don't really see Planktothrix or Aphanizomenon blooms in Lake Okeechobee, at least not from the data that I've collected. So to determine why they're blooming in those lakes, what's causing them to bloom when they're blooming, just to give us a better idea of that, right? And this can help us, kind of just gave my last point, but that can help us understand why the diversity of these bloom forming genera in the Kissimmee chain of lakes is higher and different compared to Lake Okeechobee. So I want to thank you for your attention. You can reach me here at flefler@ufl.edu. This is a QR code to the CyanoSeq database. I want to thank all of the funding agencies that helped with all of these works throughout the years. U.S. Army Corps of Engineers, UF IFAS, SNRE, Florida Sea Grant, South Florida Water Management District, FWC, FWRI. Several people helped with the Belize sampling. Of course, my lab members, my advisor, Dr. Dale Laughinghouse, and my past, present, future lab members. And thank you. I guess at this point, I will take questions if there is time. I'm not sure where we are at with time, Allison.
Yeah, so we have just 2 minutes, but there is a question that popped up a little while ago in the chat from Tina. So maybe if you want to answer that one, and then I know everybody has your contact information for additional questions too.
Okay, yeah, this is great. What efforts have been made to communicate this info to county labs and FDEP who are actively doing algal IDs on a regular basis? So when we publish these data, we plan to make these data open as well. And then also Dr. Laughinghouse, I don't want to speak too much on his behalf, but he does an algal ID course every year. I can't remember where and when this is. So I know that there are, at least I've heard there are people from the FDP who have taken this course to help with their algal identification. And I guess that's probably more a question for Dale than me. I'm not involved in that. Yeah, Hans.
Hey, Forrest, can you hear me?
Yeah.
Oh, great talk. You've learned a lot from your mentor. You guys can give a presentation quicker in the smallest amount of time than anybody I know. But anyway, I have the stuff is really interesting. But what I wanted to point out is that, of course, Microcystis likes nitrogen because it can't fix nitrogen. But and I still call it cylindrospermopsis. What do you call it? Raphidiopsis.
Raphidiopsis, yeah.
I noticed it also liked high NP. And, one thing we found out about cylindro is that it likes, it's very good at taking up nitrogen, and it won't fix nitrogen unless it absolutely has to. It's a very kind of low frequency nitrogen fixer. So it fits this nitrogen, this high NP ratio pretty well that you've, laid out. And it is an unusual one compared to the other diazotrophs. It just, it hangs out in subsurface waters, doesn't need much light.
Oh
we'll
yeah,
take
yeah,
that.
it's really interesting. So
Yeah,
we grow it here in the lab, and we can we can remove a lot of nitrogen, and it still won't produce a lot of heterocytes. It seems to
yeah,
be a
yeah,
really interesting, really difficult nitrogen fixer. So, and we
yeah.
see it when we're out collecting samples. We confuse it a lot of times because it won't have its heterocytes, so it looks maybe a little oscillatoria like, maybe a little lyngbia like. So
Right, right.
it's
Yeah.
yeah,
Yeah. Well,
it's a
great
difficult
stuff.
one.
I'll leave some time for others to ask you questions. Great talk.
All right,
I
thank you.
think that actually that puts us right up at time. But Forrest, thank you so much for a really fascinating talk. I really enjoyed that. And like I said, for everybody else, if you have questions, his contact information is on the flyer for this talk. For everybody who is registered for the seminar, please don't forget to take a look at your attendance and iFlicker, make sure all of that looks good, and let me know if there's other issues. And otherwise, I will see you back here on Monday for our last seminar of the semester, which is crazy. But thank you very much. I appreciate it.
All right, thank you.